Thank you very much for your time interrupt. Maybe I have to apologize because two speakers this afternoon from the same institutions in this very small population. Anyway, I'd like to talk about
one set of the
well, yeah.
one set of the participants are American.
Very much. Okay. So I'm going to talk about a bit a basic study on the mode of action of the bioactive compounds. So my group has long been interested in the mode of action of natural products, and so far we have identified several important biological molecules targeted by these structurally different, unique natural products. In particular, these three compounds inhibit the histone deacetylase activity, as Dr. Tama kindly introduced. And the discovery of these HDAC inhibitors opened up a new avenue of research on chromatin structure and function, and its potential to become anti-cancer drugs. Indeed, so FK228, this compound, has been approved for the therapeutic agents against the CTCL. And so far, 4 different HDAC inhibitors have been approved as anti-cancer drugs. Today, I'd like to talk about the mode of action of this compound, theonellamides. The deep mycosis is one of the important infectious disease. So the most cases are opportunity infection, which occurs when the patient's immunological activity is downregulated, such as cancer or HIV infection. So it is refractory to the conventional therapy using the antibacterial antibiotics because fungi are eukaryotes like human. So we need the antifungal agents. So currently, these 4 different types of chemicals are used clinically. And perhaps most important class is the azole, which inhibit the ergosterol biosynthesis, and the candins, which inhibits the cell wall component beta-D-glucan synthesis. So however, because of the threat by the drug-resistant strains, we need more variety of the antifungal agent with different mode of action. So that's why we are interested in this compound, theonellamide, which is bicyclic peptides derived from marine sponge, Theonella species. So these compounds show the broad spectrum of antifungal activity and with moderate cytotoxicity to mammalian cells. So because of this very different structure, we assume that it has the unique target. So we started to analyzing the target by identifying the binding proteins by using the photoactivated linker coated beads conjugated with theonellamides. So this method is very, very useful because you don't need any SAR to make a biologically active compound with beads. But I don't tell you the detail, but this method was established by Dr. Hiro Osada, the previous speaker. Anyway, this method is very, very powerful, but unfortunately in this case we couldn't detect any specific binding proteins. So you can see several bonds here, but all these are non-specific because the addition of active theonellamides like this, theonella A or theonella A, never compete with any bonds, indicating that these proteins are non-specific. So in general, for target identification, it is quite important to detect the physical interaction between query compound and the target protein, but it is not always successful because of the unstable or a weak interaction between proteins and chemical, or low abundance of the target protein in cells. So in that case, it is quite the detection of genetic interaction is quite useful. So chemical genetic interaction is based on the idea that the changes in gene activity, such as a mutation or overexpression, cause the phenotypic change. So for example, qualitative or quantitative change in the target protein results in the altered sensitivity. Okay? So several years ago, we have cloned all the genes encoded by the fissionized genome, and we could establish the expression system one by one. So by using this system, we determine the subcellular localization of all the protein encoded by the fissionized genome, which is called localized zone. So we took two different strategies to identification of the target molecule. One is to detect the chemical genomic profiling. So we have collected all about 5,000 strains in which each one of genes are expressed under the control of inducible promoter. So you can compare the fitness of yeast between induced or uninduced conditions. By this screening, we identified 32 genes which can induce altered sensitivity to theonellamides. The second approach is to compare the chemical the target visualized by the fluorescently labeled compound and localized on database. Okay. So we first analyze the chemical genomic profiling by with the target known compound like this. So to the clustering analysis shows that there is similarity between theonellamide and polyene macrolide antibiotics such as amphotericin B and nitazine, but this interaction was quite weak. So next we analyze the gene ontology terms of the 32 hit genes. So we ranked the enrichment of gene ontology and compared with target known compounds. So in this analysis showed that quite strong interaction between theonellamide and FK463, the micafungin. Micafungin is a strong inhibitor of 1,3-beta glucan synthesis. So the question was whether theonellamide also affect the beta 1,3 glucan synthesis. So therefore we compare the phenotype of these two compounds. So in the case of FK463, you can see the leakage of the cytoplasm because of the loss of cellular integrity. However, in the case of theonellamide, we can't see any sign of cell lysis, but strikingly that the cellular component was highly accumulated instead. So this signal, for instance, signal by color white is due to the accumulation of 1,3-beta glucan because the mutant in the 1,3-beta glucan synthesis failed to accumulate this fluorescent signal even in the presence of theonellamide. So we have identified these genes which are commonly affect the sensitivity to both theonellamides and FK463, but some of them, these green ones, will oppositely affect the sensitivity to different compounds, suggesting that these compounds counteract each other. So indeed, we treated the cells simultaneously with these two compounds, and what we found was that the presence of theonellamides rescued the cell lysis phenotype induced by FK463. That means theonellamide counteracts the action of FK463. So next we try to understand the mechanism of the overexpression cellular component. So once the beta glucan synthesis is mediated by the activity of LO1, the membrane small g protein signaling molecule. So we expressed the wild type LO1 or dominant active LO1. Then you can see the further increase in the cellular component. In contrast, expression of dominant negative mutant of LO1 totally inhibit the increase in the 1,3 beta glucan induced by theonellamide. These results clearly show that the theonellamide in the cellular synthesis is mediated by the signaling molecule LO1. So, okay, the biggest question is, what is the direct target of theonellamide? So to address this question, we used the fluorescent level derivative of theonellamides. So by using this probe, you can see the target visualized by the cellular fluorescent derivatives. And then, so if theoretically you can pick up the candidates of the target molecule from the database of the subcellular localization, which shows identical localization with the theonellamides. So, sorry. Okay. So, to this end, we synthesize the fluorescent labeled theonellamide. So, theonellamide body P or theonellamide alka amino coumarin. So, what you can see here is the results, and theonellamide target appear to distribute the cell growing ends and the cytokinesis region or the septum region of the cells undergoing cytokinesis. So among the database, we found a number of transporter proteins showing the very similar distribution. So including PMN1. So PMN1 is known to be localized show the polarized distribution depending on the biosynthesis of ergosterol. So this polarized distribution depends on the biosynthesis of ergosterol. So this result suggests the link between ergosterol biosynthesis and theonellamide target. So we obtained several mutants of ergosterol biosynthesis pathway and look at the effect of the mutation on the theonellamide effect. So what you can see here is some of the ergosterol biosynthesis mutants were resistant to theonellamides, and also these resistant mutant showed a lowered binding of theonellamide body P theonellamide like this. So these mutants showed lowered binding activity to theonellamide, and also these mutants also showed that lowered amount of the biosynthesis of 1,3 beta glucan. So these results most likely scenario is that the selenide target is not the protein, but a lipid molecule such as steroids. So to test this possibility, we looked at the direct interaction between several lipids in biomembrane like this. So what you can see here is theonellamides strongly binds to ergosterol and cholesterol. And we have tested several other related compounds, and we found that not only ergosterol but cholesterol, but also several other compounds can bind to theonellamide. But these compounds were inactive to bind to theonellamides. The common feature of these inactive compounds are the lack of beta hydroxy group at the carbon 3 position. So we conclude that theonellamide specifically recognize the 3-hydroxy steroids. So next we tested in vivo experiments. So by using the Philippine as the known marker for ergosterol. So as you can see here, these two fluorescent markers are very clearly co-localized like this. But if you look carefully, the region stained by theonellamide are a bit more confined region. So suggesting that theonellamide binds highly enriched, the region highly enriched with the ergosterol. So in yeast, the actin microfilament is quite important to determine the polarized localization of ergosterol. So if the temperature-sensitive mutant of actin or treatment with latranquin A is a specific inhibitor of actin polymerization, induced the inhibit the polarized localization and show the uniform distribution of the ergosterol. However, in this case, cellular binding was almost gone. Suggest this probably due to in the absence of active actin filaments, the ergosterol will be distributed uniformly in the biomembrane, which therefore the local concentration of ergosterol is lower than the level that required for ergosterol theonellamide binding. So there are several chemicals which binds to ergosterol. The most well-known, the best known is polyene antibiotics, which form the pore on the membrane. So therefore we compared the phenotype induced by both compounds. So the most well-known feature, characteristic feature of polyene antibiotic in yeast cells is enlargement of vacuole and also of acidity inside. However, theonellamide treated cells showed fragmented vacuole compared to wild type. So the phenotype is different. And also, polyene antibiotic quickly killed cells, so only 1 hour treatment was sufficient to kill the cells. However, the cytotoxicity induced by theonellamide was time dependent. So these results strongly suggest that mode of action is totally different from polyene antibiotics. Furthermore, we looked at the vacuole morphology in the cells overexpressing RO1 mutants. So overexpression of wild-type RO1 showed the fragmented vacuole morphology, which is very similar to those observed in theonellamide treated cells. And also expression of RO1 dominant negative mutant partially rescued the fragmented morphology of vacuole. These results indicate that the morphological abnormality of vacuole in cellular treated cells is due to the activation of the RO1. So let me summarize here. So theonellamides specifically binds to the ergosterol in the membrane region rich in ergosterol. And after then, the membrane key molecule, RO1, is activated by unknown mechanism, which induce the vacuole fragmentation and also overexpression, overproduction of 1,3-beta glucan in the cell wall. So amphotericin B or some other poly-N antibiotics also bind to the ergosterol, but it forms the pore which allow leakage of cellular ion or other cellular components. So the mechanism of action is totally different. So theonellamides represents a new class of cell binding molecules. However, still there's an open question as to how theonellamide induce the change in the activity of proteins after binding to the membrane ergosterol or membrane strokes. So we next examine the in vitro effect of artificial membrane using the giant unilamellar vesicles containing cholesterol. In this case, this time we use cholesterol. So as you see here, the amount of theonellamides binding to the vesicle well correlated with the amount of cholesterol, like this. So perhaps you can see, okay. So if you incubate the theonellamide with the lipid vesicle containing some other different lipids, you can see the theonellamide specifically binds to the membrane region containing DOPC, okay. So which is labeled by rhodamine DOPE, but not labeled the sphingomyelin region. Like this. So this result suggests that theonellamide specifically binds to the lipid disordered phase, but not lipid ordered phase. So this was also confirmed by other experiments. So in this case, we use different lipids. So the amount of theonellamide bind to the lipid vesicle was lower if you use the DPPC or DSPC, which form the lipid ordered phase. Okay. So and also the theonellamide treatment induce the lipid order membrane phase separation. So high concentration of cholesterol often induce the uniform only one lipid phase. But if you treat this membrane, this vesicle with theonellamide, then the phase separation occurs into lipid ordered and lipid disordered phase. So this activity was also observed in biomembrane in cells. So by using this specific marker compounds, which detect the lipid order domain, then you can see the big change in the membrane order. And simultaneously, at the same time, theonellamide induced the cell contraction like this, very dramatic. So this effect was very rapid and transient. So within 30 minutes, the cells showed strong contraction, but this effect was reversed after several hours like this. So this effect is totally dependent on the presence of cholesterol, because depletion of membrane cholesterol by using the methyl beta cyclodextrin prevent this change. So this dramatic cell shape change was also dependent on the actin polymerization and depolymerization and tubulin polymerization. But although the stabilization of microtubule by Taxol did not affect. And also, although I don't show the data today, this effect was also dependent on the cellular energy, because the treatment cells with low temperature at 15 centigrade, or treatment with inhibitor of respiration or glycolysis also inhibit this dramatic change in cell morphology. So theonellamide may modulate the membrane order, which in turn induce dysregulation of cytoskeleton. In the end, energy-dependent cell contraction occurs. So therefore, the theonellamides is quite unique compounds that induce change in the membrane environment, as well as membrane signal perturbation. So therefore, theonellamides will serve as a very useful tool for studying the enigmatic nature of cell membrane. So I'd like to thank my colleagues and my members. So most of the work was done by a very talented postdoc, Shinichi Nishimura, who is currently working for Kyoto University as assistant professor. And effect on mammalian cells was done by former graduate students, Yuko Arita. So the theonellamides, very amazing compound, was a kind of gift by Shigeki Matsunaga at Tokyo University. Thank you for your kind attention.
If you have questions, please ask. This trichostatin, your deacetylase inhibitor, does it affect also the fatty acid pathway in plasmodium, which is a non-mevalonate pathway for fatty acid synthesis? Mm-hmm. I don't know if it's trichostatin or trichosanthin. Is the same? Could be also implicated, could also affect this pathway of fatty acid synthesis in plasmodium?
Actually, I'm not sure about.
You have to give to Mr. Rosado. It's Dr. Rosado.
No, I don't think so. So trichostatin can bind the iron also, because it has the hydroxamic acid. But in the case of trichostatin, this metal binding moiety is quite important to interact with enzyme zinc ion. So of course it can form the chelator complex with free ion, but it is not so strong. Perhaps his compound is much more strong.
Thank you. Other questions? I think it's my turn, but if Dr. Castello is prepared, and are you in a hurry, you want to present? Or?
It's up to you. It will be different from the topic now,
Okay.
so I can wait for the end of the session. They proposed to me to be just at the end of the entire session.
Yeah, so I will start.
Because you know you are in the
Thank you very much. Goodbye.